Okay guys, the human ear. My name is Mr. Mpepu and today's date 1 May 2024, Wednesday, and the time there 47 minutes past 11. With that being said guys, sanibonani, dumelani, afsheni, hello, nda guys to all corners of South Africa and Africa kabuphara. I personally welcome you to my class. I personally welcome you to my session. Today we are also dealing with our human ear past examination questions. So if you are new here, don't forget to subscribe, don't forget to comment, don't forget to like, and don't forget to share this video so that we can reach more learners out there. Whether you are people pink, left, right, you don't have legs, you have legs, you don't have hands, you don't have whatever, we welcome you here. Whether you are in hospital, you are today's holiday, right? Whether you are drinking, whether you are partying, whether you are doing whatever you are doing, we welcome you here. Join us, learn life science with me. We started at week 1. Today we are at week 7, right? Under responding to the environment, humans, and then we are still under human ear with our weekly plan. Let's pass by here. Monday, Tuesday, Wednesday, Thursday, and also Friday. Monday we said that we are going to deal with the structure and function. We did this. Tuesday, hearing and balance, did this. Wednesday, question and answer, we are going to do this. Thursday, hearing defects, we are going to do this tomorrow. Friday, and then tomorrow we are also going to do the revision for human ear, right? Then Friday we are going to do end of these topic exercises covering everything: our human nervous system, central nervous system, peripheral nervous system, autonomic nervous system, nerve, reflex arc disorder, Alzheimer's diseases, and multiple sclerosis, our human ear, our cornea, our lens, our suspensory ligament, our ciliary body, our ciliary muscle, our retina, our choroid, and our sclera, everything, right? So welcome guys. If you are late here, where were you? And for mark that register, don't forget to subscribe. Last time I checked, we were at positive 130 subscribers, guys. I really like to appreciate you. I love you. Nginithanda nyani. Azishe, let's learn, right? To take a holiday, we can do this fast so that after here we can go and party. Rona will party. When now you will watch this later during the night, right? So thank you. Human ear, we are done here. We are done this part, right? So the part that we are left with, it's our hearing defects. This 2 year that we are going to do tomorrow, Thursday, then Friday, according to our plan, right? Our weekly plan. So we are moving on. Examination guidelines, done. Where? Up until here, we are done. You will agree with me, right? Agreed. Let's move on. Yesterday we did our balance. Then we have 3 in our inner ear. We have our what we call this semicircular canal, right? With our semicircular canal, they continue and form the vestibule there with our utriculus and our circulus, and then from there this is going to form that snail-like shell structure which is called the choroid. So we are moving to this direction. From this direction we have our ambula there, and then ambula they are having our cristae. Then from our cristae we are going to have our macula there, where in our vestibules, right? You still remember this. In our vestibules we are going to have a maculae, and this up there it is speed and direction, and then this side here it is position. So cram that or know that in this order. Moving from up, we have ambula having cristae reacting to the stimuli, changing in direction and speed, and then from there we move down with our inner ear. Moving down, we go to our vestibule, made out of our utriculus and circulus, right? Here they start with circulus and utriculus. Utriculus up, circulus down, right? And then are stimulated by the position of our head. Then from there you can convert that stimuli into the impulse, from impulse via the nerve to our cerebellum to maintain the balance. You understand that, right? So we are just passing by here. You are agreeing with me. Then we did this homework. I gave you this homework, right? We have done everything. Today we are just doing with questions. Fast holiday, happy workers day to all those who are working and all those who are unemployed. Let's hang in there. One day right? One day God will make miracles. Don't I even don't think like there will be people here who are unemployed, what do we call unemployed, busy following us. I believe that we are mostly grade 12 students, maybe teachers nyana there, who knows. Then number 1, we have our diagram, right? We've studied this diagram Monday, we did the structure and functions. And then with Monday, we learned that we have this side here called pinna. We have this side here called auditory canal. We have this side here named tympanic membrane eardrum. We have this part here named ossicles in our middle ear. We also have this part here named our semicircular canal. We also have this part here named ambula. We also have this part here named vestibule. We also have this part here named oval window. We also have this part here named round window. We also have this part here named cochlea. We also have this part here named eustachian tube. Eustachian what tube, right? So we are moving on. We decoded the question. We know what is happening with this question. At this moment, I know or I believe if you learn Monday, you learn Tuesday, now you know what is happening with our human ear, right? We can move on. Then number 1, write down the letter only for the part which. Write down the letter only. Follow the instruction. Follow the instruction. They want you to write the letter only, not the name, because there's no name there. Then bar number A, that amplifies the vibration from the tympanic membrane. That will be number B. You get your 1 there. Number B contains the receptor for hearing, where be number C. Number C there. You are done. You see, only letters only. Then number 2, I also told you that you can also go and try to answer number 2, but osteosclerosis is the genetic form of hearing loss caused by a stirrup becoming immovable, right? Explain how this condition may cause hearing loss. It will cause hearing loss because that stirrup there, those stirrup there will not be able to transmit the vibration, right? Vibration to the inner ear, right? 3 marks there. So when you are going to explain how this is coming to come about, because the stirrup is immovable, the stirrup will not be able to move, will not be able to exert the vibration to our oval window, and then from there the oval window won't receive that vibration, so that the vibration will go on path via our pressure wave. We call them pressure waves. And then from pressure waves, our organ of Corti, they won't be stimulated, right? So we're just passing by here. If you did this, thank you. If you didn't do this, we're just passing by. Today we'll be focusing on our previous question papers, right? So I also asked you to go and try number 4. Number 4 was the vestibular branch. And another thing that I was talking about yesterday, I said, and our auditory nerve, right? They can say here they say the vestibular branch of the auditory nerve. This is the branch of the auditory nerve. From our textbook and our study guide, I found that they are referring to our ear nerve as a auditory nerve, right? Without correcting them. You know that yesterday I was saying that auditory nerve, also another name of auditory nerve, is called cochlear nerve. This nerve here is responsible for transmitting the impulse of hearing to our cerebrum, right? But here you can see that they can branch, vestibular branch and cochlear branch, name them auditory nerve. I'm not sure. Only if you can get this, or if you don't get this, go and watch back my videos and see what I'm talking about, right? So this is the branch that transmit impulses from semicircular canal to the cerebellum. For what? For balance, you know, right? This one is for balance. Then we'll explain the consequences if this nerve, it is affected by virus. So if this nerve, it is affected by virus, we are going to disrupt the movement of impulse from the vestibular branch to our cerebellum. So this impulse here is going to be disrupt, right? We are just passing by here. It's going to be disrupt, and then after being disrupted, our cerebellum here won't be able to receive that message, so that it will send the message to our skeletal muscles, right, to maintain the balance. So the consequences of this nerve will be with the balance. You will become dizziness, you will fall, you will not be able to maintain the balance, or you won't be able to adjust the balance, right? Brought upon the stimulus of our position, the speed and direction in our vestibular branch of this nerve, right? Semicircular canal and our vestibular there. So you understand, right? Also yesterday we did this PET test, OCAC. We did this yesterday, right? So you know this one is the pathway of our hearing. We have our pinna there, we have our auditory canal, we have our tympanic membrane. From there, this vibration is going to vibrate our ossicles, our hammer, our anvil, and our stirrup. This vibration here is going to be amplified and is going to vibrate our oval window inside our cochlea. Then when we are inside our cochlea, we see that the pressure waves here, they are going to stimulate our organ of Corti. Then organ of Corti convert this stimuli into the into the impulse, and then the impulse is going to be transferred via our auditory nerve, then from our auditory nerve straight to our cerebrum to be interpreted. I hope you understand that. Today we are moving to our basic education website, sourcing all those question papers, and come and see how we can answer this question and how they are asking this question. Thank you for being here. Thank you for choosing us. Thank you for tuning in. My name is Mr. Mpepu. Nothing much to offer but this. We start with 2021, 2022, 2023. Last question papers, right? Last examination or final examination, right? So let's start with our 2021 first. 2021 there, and then with our 2021 we can go and scan our question paper, right? I was scanning them. I find those questions that they are related to what we are studying, right? And then we have the grommets there, but the ability of lengths. Also this one 1.1.2, you know, to write everything you have to come here and write section A, and you also have to come and write question number 1. If you are going to answer question here, right, then you are also going to indicate 1.1. Then the question that you are going to answer is 1.1.2. And then the question about the ability of length of the ear. This is the ability of the length of the eye. Sorry, we did this, right? The ability of the lens of the eye to change its shapes when viewing an object that is near or far. It is what? Accommodation. Right? You'll agree with me, this one. This one will agree with me. Now, we did this. We are moving. Fast. 1.1.3, which one of the following? The visual defect. We also did this. 1.1.3, we're going to answer it. But which one of the following is the visual defect that results from the uneven curvature of our cornea? Which one? Astigmatism. That will be number D. Sorry. Number D. I want to write number D. Astigmatism, right? So astigmatism, this is what you will need to explain. Then 1.4, the receptors involved in the maintaining balance. Yes, we also did this. 1.1.4, but the receptors involved in maintaining balance is or they are organ of coating. No. Maculae only, yes, but we also have maculae and cristae. So this one will be the one. This one will not be the correct answer. So we are going to choose number C, because it's the cristae and it's the maculae and cristae that are maintaining the balance. 2 of them, right? 1.5, which one of the following structure are involved in the maintaining balance when there is a change in the direction of the movement of the body? We also did this, right? 1.1.5 there. 1.1.5, which one of the following structures are involved in the maintaining balance when there is a change in the direction? We want the direction there of movement of the body. We know we have 2. We have speed and direction, and we also have position. These 2 and these 2, right? Here ambula and inside cristae, and then here vestibule inside maculae. So we can go with semicircular canals and cerebellum. This one, right? Semicircular canal, you can see we are dealing with the direction there. And then where do we find the cristae? We found them in semicircular canals on ambular side. And then the impulse is going to be sent to our cerebellum. Then we can see Eustachian tube, no. Semicircular canals and cerebrum, no. Cerebrum, no. And then here Eustachian tube and cerebrum, no. And then the answer there is going to be number A. I hope you understand this, and I hope you are getting this, right? Then 1.1.6, but which of the following structure are protected by meninges, brain and spinal cord? Let's just go and see the one that is going to say the brain and spinal cord. Number B, brain and the spinal cord. That will be number B, right? Then number C, we haven't do this diabetic, what. Then 1.8, during a reflex action, we also did this. We can also answer it. 1.1.8, but during a reflex action, impulses enter the spinal cord by means of, they enter there. We have the dorsal root. Let's see, our sensory neuron, yes, through the ventral root, no. Number B, sensory neuron, yes, through the dorsal root, yebu, number B there, right? You'll agree with me. Then number 1.1.9, this one ADH, we haven't done this. We are also going to do this next week, right? Then 1.2, do we have anything here? The release of ovum, the microscopic gap between, yes. We are also 1.2. We see another one, 1.2.2. We see this one, the microscopic gap between the 2 consecutive neurons. This one is called synapses. Synapse, right? And then number 1.3, the period develop, no. The growth movement plants, no. We haven't done this. We are going to do this next week. Next topic, right? And then the type of development of beds, no. The extra embryo, no. The tubules in that, no. The plant hormone, no. We are only going to secure that 1.1.2. 1.1.3, do we have anything? Let's come this side and look. Corpus gland, no. Cranium, this one, yes. And this one here. So we are going to back this 2 2 2 4, right? This one is 2 4, 1.3, we go and indicate. Then we want to answer 1.3.2. 1.3.2, a component of the peripheral nervous system. The spinal nerves, yes. The spinal cord, no. This one is not the component. This one is part of our central nervous system. Then answer there is going to be A. Don't forget A only. And then we go to our 1.3.3 with 1.3.3, but this is the disorder of an nervous system characterized by degeneration of the brain cell. Degeneration, we know that this one is our Alzheimer. They are degeneration or they are dying, right? So that was going to be B only. Where now you secured that 4 marks here. You can see it's 3 multiplied by 2, 1, 2, 1, 2, 1, 2. So when now you'll get this 4 there, right? We are moving on fast. It's holiday. I also want to go out and do some things, right? And then here we have our what we call our, you did this term 1, right? You also have to, one thing again, you also have to master this, which one is paper 1, which one is paper 2, right? So these questions here, they're also going to make you understand that this one here is paper 1. Our human reproductive, male and female, paper 1, right? We did this term 1. If you forgot this, we're also going to do revision for this if I have time. And then also this one, we also do this term 1, right? Our ovum and our sperm, our tail, our acrosomes, everything like that, right? So we are scanning. We also did this with me, right? So we are going to do section B, question 2. You indicate this question. Now if you are following me day by day, you know what I'm talking about, or you know what I'm doing here, right? So if you are not following me, my guy, go back. I still have time to secure all those marks that we are securing. So you can see here we are securing number 9. We did this together, right? You'll agree with me. Then here, this is the thing, 2.2.1, 2.1.1. As keys, yes, we have the neuron, right? 2 neuron. We decode the question. Before you can answer, you decode the question. As you can see, the diagram below represents the type of neuron found in our human body. They didn't tell us the name of the neuron. Let's expect us to be asked which type of neuron is this. And then we can see this one is not myelinated. So the speed there, you are decoding, right? And then here we have the dendrites, we have the axon, and we have the axon terminal. You decode, right? So after everything we are done, we can move on to our question. Question number 2.1, identify the type of neuron shown. You see what I was talking about. Then the type neuron here will be the motor neuron, right? The last motor neuron, right? The last neuron there, that we can see that this is the part neuron. The sensory neuron will be like this, and then the interneuron will be like this. This will be our cell body in our interneuron. You will agree with me like this, right? Then this one will be our motor neuron, seeing our cell body here connected to something going down there. You'll agree with me. 2.1.2, but use the letters A, B, C. We are only going to use this letter only. Give the correct sequence for the transmission of an impulse along the neuron 1. The neuron 1 will use only ABC, and then the neuron will start from our cell body to our axon like that. So we'll start with number C, then the answer will be C, B, A, right? You'll agree with me. You are 2 marks there, using letters only, right? Then 2.1.3, if you don't agree with me, comment that I don't agree with you, Mr. Mpepu, here and here and here, this is wrong or this is correct. Or I think the best way is to say, Mr. Mpepu, question 2 or section B, question 2, 2.1.1, I think the answer is this and this, because you might find that you are the one who's mistaken or I'm the one who's mistaken. So let's use the proper words or the proper interaction, I think. Then 2.1.3, but explain how the speed of transmission of impulse will be differ from the neuron 1 and the neuron 2, right? Neuron 1, neuron 2. So when now you'll come and indicate that neuron 2, not myelinated, right? Myelinated. And then neuron 1, because this one is what is going to make you to understand, to explain here the speed of transmission, right? Where are you saying that the speed of transmission will be fast here and slow here? Hence that is the answer, right? But you can see it's 3 marks here, so you have to also to come and give the basis, like educating a marker, as if like you are educating him or her. Then neuron number 1, myelinated, right? Then you'll come and say the neuron 1, neuron, but explain how the speed of impulse will differ. Neuron 1 will be, the speed will be, or the speed, you'll say the neuron 1 speed will be faster due to myelination, and neuron 2, the speed speed will be slower due to unmyelination of the neuron, right? So you see the basic what we are doing, or how am I answering this? Where do you see this? You see this here, right? I also did this when I was doing neuron, saying that they will give you a question saying that the one is myelinated, one is not myelinated, right? So we are moving on fast. 2.1.4 there, but 2.1.4 explain why a person will feel the stimulus but will not be able to respond if this type of neuron is damaged. Then yes, we want to explain why the person will feel the pain. Will feel the pain because we have the sensory. Yes, what we have to explain, we have the receptor, sensory neuron, interneuron, motor neuron, and effector. So we're not explaining this. You'll say that the person will recept the pain, and then the pain or the stimulus here, not it's either the pain or what, but the stimuli. The person will receive the stimuli, and then the stimuli will be passed to our central nervous system, and then with our inside our central nervous system, another neuron is going to be interpreted, and you're going to feel the pain, right? But it will not be able to respond. The person will feel the stimuli. You are going to feel the stimuli. Here we are referring to stimuli. It's not the pain, right? So you are going to feel that stimuli, and then because our motor neuron, it is cut there, or the motor neuron, it is you see that the damage, right? The neuron is damaged. If the neuron is damaged here, the impulse will not be completed, or the impulse will not be sent successfully to our effector, so that when you will respond, right? 3 marks here. So this is what you have to explain, right? The message will you start here. The message will be recepted. The stimuli will be recepted, sensory neuron to our central nervous system to be felt, right? You will feel it. And then another thing, this one, they are saying that you will feel it, and then from there, while this impulse has to be sent to our effector for you to respond, because the motor neuron, it is damaged, you are not going to be able to respond because the impulse will not reach our effector there, right? Same that side. So we move on, we scan our question paper for another question there. This will be another question. Fast 2.2 2.2, our human eye, right? We did this. Our human eye, we have 3 layers, as you can see there. We have 3 layers. The first layer will be the sclera, the second one choroid, the third one retina, even if it's not there, the number D fovea, or our yellow spot, blind spot, optic nerve, and then suspensory ligament, our lens, our iris, ciliary body, ciliary muscles, cornea, aqueous humor, vitreous humor. Then what else? What else? We are done with our basic parts, right? So we go to our 2.2.1, right? But identify part C. Part C there, it's our choroid, right? You can agree with me, choroid. You'll agree with me, right? Then 2.2.2. Then with 2.2.2, but give one function of part E, where is part E, our suspensory ligament, is to hold a lens to place, right? This one is to hold the lens. This is the function. It's 1 mark. Give one function of part A, 1 mark, right? Or to regulate the shape, right? You can, whatever you want to say. Right. Whatever, betunana. Whatever, give one function of E, our suspensory ligament. What is the function of suspensory ligament? Is to hold our lens into place, right? Hold the lens into place, or to regulate the shape, or to function in our accommodation, to stretch the lens, and to whatever, right? Whatever function that you can come with here, only 1 mark. Or you can just say this one function in our accommodation to cause our lens to be less convex, or stretch there, so that our lens will be less, or will be flatter, will be less convex, or relax, so that our lens will become more convex. Either way, you are killing this cat, and make sure that the cat, it is dead. 2.2.3, but state why the clearest image will form when light rays fall in part D. Part D, because this one contain what? Contain more cones. Write down 1 mark. Don't have to say much. 2.2.4. Then with 2.4, Barry, let's keep it there. 2.2.4, explain one way in which part B, which one is part B? Part B, this one is clearer. Explain one way in which part B is structurally different from part F. Part F, our lens. What is the difference between our sclera and our lens, right? you come here to 4 marks anyway. You are going to say the part B. You start by saying part B. This one is a sclera like this, and then you are going to explain the sclera made out of the sclera is not transparent, right? Either way you want to say this, the sclera is not this one, this is not transparent. They said they want the structurally, right? Structurally, how is it structured? It is structured in a way that it is not transparent. Transparent. While part, you agree, is 4 marks. So you want to do the different that will apply to B but not in F, and also something that will apply in F but not in B. Here you are going to say this part is not transparent, and then part F there is going to be our lens, and the lens, it is transparent. 1 mark, 2 marks, 1, 1, 2, right? Part B, sclera, this is not transparent, and then part F, this one, it is lens, right? And then this one, it is transparent, right? Transparent. And then another one, you are going to say part B does not allow the light to pass in. How other way? Part B does not allow the light to pass through. Although it might look like the same answer here, because if you are saying something is not transparent, that means that something cannot allow the light to pass through, right? Then you can also come and say number F, allow the part to pass through. So let me not repeat myself. Let's find another one. We are going to say part B, it is inelastic. We learn about this, right? Part B, we are going to say part B. Here I was repeating myself. Part B, it is inelastic, or it maintains the shape, right? But part F, it can change, right? One can change shape. This one can change shape. You know this when we are doing accommodation. The part F is a length, it can stretch or it is elastic, but number B, it is inelastic. It cannot change, it is rigid, it also curve, it also make this eye of ours to maintain the ball shape, right? Then part F there, this one is our lens, and our lens there, it can change shape. We agree, are we together? Yes. Let's move on. Then we go to number 2.2.5. But describe how the muscles in part A. The muscles in part A. Where is part A? Part A, this one, it is our iris, right? Pupillary mechanism. Describe how the muscles in part A function to increase the amount of light entering the eye. While we are entering the eye, we are increasing, right? So we are going to say 2.2.5. We are going to say circular muscles, So for this to increase, circular muscles have to relax. Circular muscles will relax, right? And then another part there, we are going to have our radial muscles. Radial muscles will contract. I agree, we want it to open, right? Will contract. You'll agree with me. Then the last part, more light will pass. More light will pass, like that, right? To describe the muscles in part A function to increase the amount of light entering the light. And then here we can, rather than saying more light will pass, we can say the pupil dilate, right? Dilate or increase, right? So we want that pupil to increase while this is happening. They also want us to explain the mechanism of pupillary mechanism in a dim light, as you can see, right? You agree with me? 2.2, 2.2.6, then 2.2.6. But describe how a blurred image forms in a person with normal vision wears a spectacles with a biconvex lenses while reading a book. Then here's the thing. The question in your describe how our image, we talked about this. When you take someone's spectacles and you wear them, you are going to see a blurry vision, right? When you take someone, so here the thing, the biconvex, what is the biconvex doing? The biconvex, we say these are there like this, so they are going to form more light, they are going to cross more light like this, right? So here's the thing, when now tell us what happens, describe how this blurred image is going to be formed when a person with normal vision wears a spectacle. So the person that has normal vision, when they wear the spectacles reading the book is also is here, and then you know that with accommodation our pupillary mechanism is going to be big like this. Our accommodation, our lens is going to be big, to becoming more convex. So what when you are doing, you are making this eye to be more convex. So what is going to happen, this one is already refracting that light, but because when you are wearing a biconvex, you are going to refract that light more. That light here now falling, or that image now forming in front of our retina. So that is the thing. You have to describe that, right? So how you will describe that, I'm not sure. You are going to say the biconvex spectacles, or the spectacles, biconvex lenses or biconvex spectacles will refract light more, right? Will reflect more, or will reflect light more, right? Then this resulting in the image formed in front of the retina, right? Causing or resulting in short sightedness, right? So this one will form, this one will cause, this one will result in this image there blurry, right? This one will describe how the blurred image forms. So this one will resulting in blurred image formed in our retina, or yellow spot, right? So we agree, right? This is what we are explaining. This is how you can try and kill this cat this side. So let's see another thing. If we also have another questions here, you can see that these questions here, they make up many questions here in 2021, right? We have elapsed our 30 minutes just discussing 2021 question paper. As you can see here, reproduction term 1, right? And also you can see here mixing our nervous system and our endocrine system. We haven't talked about this. With our nervous system, there we have our cerebrum, our corpus callosum, or medulla oblongata, and our cerebellum. Know those parts, but identify part A, cerebellum, state 1 function of part C, to join or to allow the impulse transmuting from our left and our right hemisphere. Simple as that, right? So from there, we did temperature. We're going to do this next year. Our responding to our environment by plants, we're going to do next topic, and then we are done with 2021. Let's rush to our 2022 fast. 2022 fast. Let's see questions. Can we make this on? Yes, we can. Then but various options are provided. We are choosing same thing. Section A, question 1. Don't forget, if you are going to answer those question 1, right, then you come 1.1. Let's see which one of the following maintains the shape of eyeball. We did this 1.1.1.1. Which one maintain the shape between the retina? No. Vitreous humor? Yes. Lens? No. Cornea? No. Then answer will be number C. Then the choroid? Yes, we also did this 1.1.2. The choroid is richly supplied by blood vessels. Yes, number A. That one don't even have to talk much, because contain photoreceptor? No. Refract light? No. Send impulses? No. 1.3, which one of the following occurs immediately? Fertilization. We didn't do this. On a hot day, we are still going to do this. The normal site of fertilization of female, we didn't do this together. And then we move on. But which one of the following best describe the events of accommodation? 1.1.6, right? Challenging Nyana there, 1.1.6. But which one of the following best describe the events of accommodation when a person is viewing an object that is less than 6 meters? Less than 6 meters, we want our lens to become big. And then from there, our suspensory ligament, for this to happen, we want them to slacken. So we can see here they give us suspensory ligament. We want them to slacken like that, right? These two options, they stand the chance to be the correct one. And then the tension there, we want it to decrease also this side. Then the answer, we're going to see it. Suspensory ciliary muscles, we know that for this to happen, they have to contract there. So the answer will be number B, right? Thank you. Then we come to this 1.1.7, then 1.1.7, 1.1.8, they are related to this. Which part of this, or which type of neuron is this? Sensory neuron, right? You can decode this question. Yes, we have our cell body, we have our axon, we have our myelin sheath, we have our dendrites, we have the impulse direction, we have our terminal, and then we have our axon synaptic knob. Then 1.1.7, the axon is represented by structure 4, number D there, right? 1.1.8, but which label part affects the speed of impulse? Myelin sheath, number 3, number C there. You see? So we move on. Then we come to this one is a response to the environment by plants. We are going to do it next week, right? Then we come here to our question. Let's see if we can get something here. The part of skull that protects the brain. Yes, 1.1.2, 1.2.1. Which part of the skull protect this? The cranium, right? Cranium, right? And then the homeostasis process, we haven't done this. The visual defects characterized by cloudy lens, we did this 1.2.3, which will be cataracts, cataracts, right? Or cataract. And then 1.1.4, the blood vessel that transport the oxygen, we didn't do this together. The part of the brain that controls body temperature, we also did this, but we are also going to do this one will be 1.1.5. This one will be a hypothalamus, right? We did this, the one that did body temperature, hypothalamus, right? Then 1.1.6, the branch of nervous system that is made out of a spinal cord and a cranium. This one is coming for the second time. We did this previously when we were doing questions somewhere somehow. This part here, peripheral, right? Peripheral nervous system, right? We did this one is coming for the second time in our class. 1.2.7, 1.2.8, finger-like projectors developed from outer membrane of our embryo after implantation. We are not going to do this. And then the hormone that regulates salt, we are also going to do this. The fluid that protect the development fetus, again with what. Then the area of the retina that contains the highest concentration yellow spot, right? This one will be 1.10. We did this. This one is called yellow spot, right? So we are moving on. We come here and see, we decode this side. Synapses, we know this. Bapstic acid, gabalance, we don't know this. You see, we are also still going to do this next week. Testosterone and osteosis, we did this previous, but the hormone that stimulate puberty, both of them here. Then 1.1, but we didn't do that with me, right? So we are going to pass it. 1.3.2, we are only going to collect these 2 marks. But the functional connection between the 2 consecutive neurons, this one will be synapse, A only, right? You collect that 2 marks and then you move on like that. The sniper 1.4, we come to this 1.4, we see that we did this. The diagram below shows the condition of the eyes for different light intensity when viewing the same object. You can see here, you can see the pupillary mechanism, you can see the pupillary mechanism, right? Diagram 1, diagram 2, diagram 3. So let's go and answer this question. This one is dealing with pupillary mechanism, right? And then 1.1.4, 1.4.1, but give the letter and the name. The letter and the name, 2 things, right? Of the part that A, you say number A, that contains muscles. That contains muscles, that will be number B, and then that will be iris, right? And then number B or number B there, but it is made out of tough white fibrous tissue. Even though we don't know, we only have A, B, C. Then C is pupil. Is pupil made out of tissue? No. B, we have already answered it. Then the answer there is going to be A, and then this one will be sclera, right? If I'm making a mistake, guys, don't forget to comment and allow me to correct myself, right? Then 1.4.2, but which diagram 1, 2 or 3 represent the eye of the person? They want you to only indicate 1, 2, 3, answer using these 2, 3 numbers, right? But which diagram represent the eye of a person in a very, very bright area? It means very bright, you have to close that, right? More light, close. Less light, open. So here we want the very bright area will be number 2. So we just come here and write number 2. Right, as you can see, the pupillary mechanism between the 3, this one is very, very much small to accommodate less light, right? The number B, where the rods are stimulated the most, that will be number 3. Why? The rods here, you can see that the rods, they are stimulated in a dim light or in a dark place, right? We learn about this one helping us to distinguish between the dark or the black and the white, right? So you can see in our 3 there, the place here or the condition here, it's dim. So what is happening is that these rods, they are going to be more activated. I hope you understand that, right? Then 1.4.3, nothing complicated here. Simple marks, we are just collecting. Which muscles are, number A, which muscles are contracted in figure 2? With diagram number 2, which muscles are contracting? We know we have 2 muscles. We have the radial and the sacral muscle. So which one is this contracting here so that our length to become small? It's our sacral. So you go sacral muscles, right? Muscles. Then number B, Number B, they were relaxed in figure 3. Where is figure 3? Figure 3 here, this one is big and the small. Which one is relaxing here? Also circular. Circular muscles. So you see our 8 muscles, we get it there. So we move on fast. Let's scout. This one is called scouting for our answers that we can answer. Here we haven't done this. We are going to do this next topic, right, or next week, if I can put it like that. And then you can see also they also brought our human reproductive male, right? So the female one, they even produced it 2021, we didn't see it. 2022, we didn't see it. So we can expect it to see in 2023. If it's not there in 2023, we'll expect to see it now in 2024, because this one they are bringing it for the second time, right? You can agree with me. Even the sperm there, you can see one, you can decode this one. You can see this one is tall, this one is long. Obviously they are going to ask you a question. Let's go. Describe the role, explain the organelles found, explain 2 reasons why sperm 1 is structurally better suited for fertilization than struct 2. You can see from this question, this is the question that they are going to ask. The moment you see a question, make sure that you can take out the question, or you can make the questions now yourself, so that when they ask you in the questions here, you are prepared. I only saw the tall one, the long tail and the short tail here, and thin thing. I found out, or I thought, or I predict that they are going to ask this question related to these 2 things. And there's the question. The first one will be with our tail there. The tail is longer, this one is short. This will be our first point. And then our next point will be these acrosomes. The sperm number 1 is contain our chromosomes, and then sperm number 2 doesn't contain that acrosomes. So these are the 2 reasons why sperm 1 is structurally better suited for fertilization than sperm 2. The sperm 2, this one, yeah, it's just wasting the time. The production of this sperm, we are just wasting our energy, because it cannot even go and break the ovum there. You know this, you learn this with your previous term 1 teachers, right? So let's go on. Here we are dealing with our hormone, puberty and menstrual cycle. You'll agree with me. And then this one, we are dealing with the fertilization, types of fertilization there, maybe the external and internal. I will agree, or I will think so. And then this one here, we are dealing with our hormone. We are going to do this next week, right? Homeostasis there, right? Then you can, the gland 1, the gland A, this, and then at the end of the day, should be able to decode this question after me dealing with you. After me cooking you, right? So question number 3, you can see we have this answer here. We did this, we have been doing this, our human brain, for many time, right? Many brain, the part in the center, corpus callosum. The part down there, this one will be our spinal cord, and this one will be medulla oblongata. As you can see from this question, they are only going to ask you questions related to this part here. So if you cannot decode this question here, you are going to have a problem. Example here, but identify part A, corpus callosum. But 1.1.2, 3.1.2, explain why a person may die if part C, it is damaged. Part C, medulla oblongata. What is being controlled here in medulla oblongata? Breathing and heartbeat. So where now there's a possibility of you to die if this part here, it is damaged, because this part here control the valuable or the vital system in our body. It controls the very much important movement, or the very important functions in our body, heartbeat and breathing. So if you get damaged there, you are going to be damaged with our breathing, and then the person will die, right? And then 3.1.3, but part B is damaged. The part B, where is part B? Part B, the spinal cord, is damaged in a person's lower back down there. And then but identify part B, spinal cord. You are going to get this one. But explain why the person will have no control of the skeletal muscles of the legs. You can see that these legs here, this is the person, the spinal cord is damaged. The person, but explain why the person will have no control of a skeletal muscles. You know that the skeletal muscles will also have our reflex arc or reflex action that it's conducted here on our spinal cord. So if it is damaged there, you are not going to have this reflex action. You are not going to quickly react. And another thing, you know that our spinal cord function as our a path, so that it can transmit the message to our brain, and then the brain, it also have to be transmitted from our cerebrum and our cerebellum back to our lungs there to maintain that balance. So the person, if it's damaged or spinal cord, the nerves will not reach our legs, so the person will end up being paralyzed. I hope you understand that, or I hope you learn that, or I hope you'll be able to explain that, because we've been explaining this for many times, that if you cut your spinal cord there, you are going to be paralyzed down there, because we also have the cerebellum there that is coordinating the skeletal muscle. So if when your legs there, they cannot receive this message from your cerebellum, there's a possibility of you to be paralyzed. Paralyzed, you have no control. I hope you get that. So let's scout for another questions. But the table below shows the recorded number of several brain injuries in a different region. There's the which region has the smallest number of several brain injuries. Just go and check the numbers there. Africa, Europe, Eastern Cape, the East London, East Mediterranean. You can see that this one, Africa. You see this one, I don't have the time. This one, Africa. This one, the region has the smallest number of severe brain injury. It will be Africa there. And then we'll explain why this data may not be accurate for the region name in 3.2.1. Why is this cannot be different? Because also another thing, the medication here they will need you to explain based on your understanding, right? Based on your reasoning capacity, right? Because they didn't give this somewhere. That the table below shows the recorded number of several brain injuries per 1,000 people per year in different regions. Then we have Latin America, we have USA and Canada, we have East Mediterranean, we have Europe, we have Africa. But the number of several brains in Africa is lowest. So explain to us why this may not be accurate for the region named there. Why this cannot be accurate in Africa? Because in Africa we don't have more medication, or we don't have many ways to understand, to find these numbers here, right? We don't have means of our medical systems to detect this brain damage here, these severe brain injuries, right? So those things, that's the things that you want to place here. You can go to memorandum. If we were to go to memorandum and see this answer here, right? Don't have memorandum there, but draw a bar graph to represent the data. Let's do this. 3.2, I still have time nyana. 3.2.3, this one here, 6 marks, right? But draw a bar graph to represent the data in the table. These 6 marks, you have to bag this. You have to take these 6 marks. So what you are going to do is a bar graph. You know the bar graph, it's like this, right? So you'll have your this x-axis and y-axis, and then from here you have to come and label this one. This one will be our independent like this, right? So here we'll have our you can say Latin America, and then here we have our USA and Canada, and then here we'll have our East Mediterranean, and then here we'll have our Europe, and then here you'll have our Africa. You write everything, right? Then this side you'll have the scale. You'll need the scale this side. Let's erase here. You'll need the scale. So if I can put the scale here, I have 800. Maybe I can use 2, 4, 6 yet. I can use 200. If you can say 2, 4, 6, 8, 10, 12. So this one is 200. 2, 400, 4, 600, 800, 1000, and then 1200. This one can, and then from there we go up and they 1400, right? For this part here, you can see that we also have this part. Then this side here, you write this one will be number of severe brain damage, right? Brain injuries. Then you write in per 100. You write everything here, right? You'll agree with me. Per, you write per 1, 2, 3 people per year, right? Then from there you come this side and write regions, regions of the world, right? Then you come and give the topic there. You're going to do the topic, the head, the table there, and then recorded. You say the number of severe brain per 100 per in a different. You're coming and copy this. Just come and copy this. The recorded or the number, you just come and copy that. The number of several brain injuries per 1, 2, 3 people per year, right? Per year in different referent regions. I want to create the space regions of the world. This one also, you're also going to get the point for this. You're also going to have the mark for this topic here, or for this heading there, right? The table's heading has to be there. Then from LA you go, LA it's 900. Where will be our 900, 800? This will be 900 here. Is the bar graph. This is the bar graph. And then you can shade it, or you can do whatever you want to do. And then USA 1.3 will be somewhere here. Then you go and mark that 1.3. And then if you want to differentiate, you want to do the differentiate. And then East Mediterranean 890. This is 800, and then this one will be 900. So 890 will be somewhere here. It will be before this 900, right? Before 900 but closer to 900. So I will want you to see it here, right? Before that. Then if you want to decorate, you decorate it. And then East Mediterranean, hey, we were at East Mediterranean, we want to Europe now. Europe is 1010. 1000 here, 10. This one will be 1100. Then this one will be 1010. So you come here and you do this, right? You'll agree with me. And then Africa 800. Where is Africa? Where is 800? This is 800. And then you come here and do this. Then you come and collect these 6 marks of yours. Where 1, you'll agree with me, right? Where 1, 2, 3, 4, 5, 6. The correct naming of your y-axis, the correct naming of your x-axis, the plot there and the plot there. The graph is bar graph and the topic 6 marks. You bag it all. Agree? Agree? We can move on now. That one was another one that I would want to end. We still have another one. My daughter. 3.3. Fast. The diagram below represents the part of human ear. Decode the question. We have semicircular canal. We have our cochlea. We have our round window. We have our eardrum, pinna, auditory canal, even though they are not going to ask this. As you can see, our hammer, our anvil, our ossicles, our anvil, our stirrup, this make our ossicles, our ambula, our vestibule, utriculus, circulus, all those, right? So, but they are not going to ask them if they are not there. So let's see 3.3.1. But identify part C. Part C, there it's cochlea. It's cochlea, right? Then something like that. 3.3.2, but state the function of part A. We want the function of D. Where is D? I can erase this, right? So that will be much clear. They're not going to ask those that with the part B. The function of part B to reduce to release release or reduce, right, pressure. Where? Pressure from cochlea or from part C. I'm not sure if that, but it is to reduce the pressure from cochlea or from the inner ear, right? You'll agree with me there, right? Then number B, move fast. Number B, the receptors found in C, those one are organ of Corti. Then number 3.3.3, but explain why a build-up of wax, of ear wax at part A, this one, may result in temporary hearing, right? So the wax will result, or the wax will block, the wax will block the sound wave, resulting in part A. You don't write part in, resulting in part A not vibrating, or you can say not vibrating enough, right? Vibrating enough. They say how many marks? 2 marks, right? Temporal. In vibrating enough, this resulting in impulse, in stimuli not reaching our cochlea, or our organ of Corti. This one will result, by explaining this, you will show that they will result in a temporary hear loss, right? A hearing loss, right? So 2.3, 3.3.4, 2.3.4, a grommet is a small device. We are going to talk about this tomorrow, right? Allow me, will come and answer this. 2.3, because now we work a grommet. Then grommet is just a small device that allows the air to move into the middle ear, and this prevents the build-up of a middle ear. Explain how the use of grommet in the treatment of middle ear affection prevents the hearing loss, right? This one is going to remove those build-up, or those liquid that build up in our middle ear. We are going to talk about this tomorrow. Allow me. 3.3.5, but describe how the receptors in part B. Where's part B? Part B, cristae, right? This one will be dealing with cristae. Crista for what? For speed and direction, right? Then buddy, describe how the receptors in part B are involved in maintaining balance when there are changes in a speed and direction of the movement of the body. Describe it. One, the cristae, they are going to detect these cristae. They are going to detect the stimuli, those stimuli that they are talking about, those stimuli, which will be the speed and what, and direction, right? After doing this, what is going to happen? Convert this stimuli into an impulse, right? So the impulse is going to be sent to the cerebellum, hence the cerebellum. Now cerebellum maintain the balance, right? Or cerebellum coordinate that balance, coordinates the balance. This is what you have to say, right? 4 marks there. It means that 1 mark you're going to be given by knowing that the receptors there are cristae. Then another mark, those cristae, they are going to react, or they are going to detect the stimuli, which is that stimuli. That one will be the speed and direction. Then after doing that, what is going to happen? They are going to convert that stimuli into the impulse. Very good. And then from there, that impulse, what is going to happen? That impulse is going to be sent to our cerebellum to coordinate that balance. That 4 marks, you can get it, right? Simple and so. Then we can scout wearing a mask. This one is not for us. We didn't learn anything to do with wearing a mask. And then read the extractive auxins next topic. So we are done. 2024. I bite more than I can chew here, or what I can swallow. But let's do this fast. It's Friday, it's holiday. It's not Friday, man. It's holiday. Then we are moving on. Same thing. Section A. Oh, we did this. Yeah. Likely we did this, right? This question, this 2023, we are not seeing for the first time, right? You still remember here we did the iris. If you still remember, if you are following me, the umbilical cord, you're following me. We did this, right? This one we are just passing by because we did this, and then what we didn't do, it was 1.1.8. The person suffering from long-sightedness, we did this, right? Long-sightedness, you see, you don't see the near. The eyeball is longer than normal, no. And then another thing is like this, right? And then the eyeball is longer than normal, no. The light rays falls behind the retina, yes. This one will be B. So we are passing, we did this. You still remember this, right? You still remember this, we did it. If you are following me, you'll remember that. So here we are done here. We did these questions. We also did this when we are saying the optic nerve, then the auditory nerve we are going to do. You still remember when I said that auditory nerve we are still going to do it tomorrow or the next week, right? When we are choosing this, and then the answer was optic nerve only there. Then we can move on. Today, let's do this. This will be our last question, right, of the day. Since you can see going down, we did this when I was talking about the dog style, right? You did this, you remember. You can see all of them. 2021, 2022, 2023, we were dealing with the part of our human male reproductive system. So we will agree, or we can prepare for final year 2024 with our human female reproductive system. This time, no, the female reproductive system, that one will help you more than anything, because you can see that this final, you can see the brain. We did this brain, and then you can, the only thing that we didn't do in this question paper is that human ear, right? You'll agree with me. We even did this Alzheimer's diseases. You'll agree with me. And then we haven't done this. We are also going to do this next week. And then we haven't done this. We are also going to do this next week. And then that's it. Responding to our plants, responding to the environment by plants. So let's do 1.4 and we are done. 1.4, we are done. We call it a day. We go and party. Holiday. Those who are shopping, go and shop. 1.1.4, but below diagram represent the human ear. Decode the question. Ossicles, semicircular canal. You see the same thing. They are asking the same thing. Cochlea, eustachian tube, round window, tympanic membrane. So they are going to ask you, go based on this. 1.1.4. So we can move on to 1.1.4. But identify parts. Now we want the identify parts B. A, we want to say 1.4.1. We are answering A, and then they want me to identify part B. Part B, semicircular canal. Semicircular canals. And then from there, number B, we go to number B. They want number E. Number E, where is number E? Round window. Those who are from Mpumalanga, God's window there. 4.4.2, right? But give the letter, they want only the letter and the name, right? Two things of the part that is filled with A. Number A, they want the letter and the name of the part that is filled with A. Between this, letters. Here's the thing. You come and see these letters. You don't come and place your letters. You can see letter A is ossicles. Are ossicle covered with air? No. Number F, tympanic membrane. No. Number B, no, because liquid. Number C, no, because liquid. Number E, no. And then we are only left with number D. So the answer is going to be number D. And then the name of that, it is Eustachian what? Tube. Right? So number B there. But it contains the organ of Corti. Number C, which will be our cochlea. Cochlea. Right? Know that. 1.1.4, 1.4.3. Then but give the letter, the only one the letter. Don't give them the name, guys. Don't give them the name. Give them the letter, because we don't have names here. This these pictures here is showing something that is not labeled by name. It's only you. They're only using letters. Hence, when I was answering something, maybe they will ask you, a letter F do what, or name the function of letter F. When now you can come and say the letter F, it is tympanic membrane. You answer this like this. You don't come and say number F, tympanic membrane, because there's no tympanic membrane there. So 1.4.3, give the letter of the part where grommets are inserted. Number F, the one I was talking about. We are going to talk about this tomorrow, and what is the function of this grommet, and what this grommet do, and how this grommet help us. And then, but it is inserted here, right? And then number B, the amplifies vibration, the part of that the letter that amplifies vibration. Number A. Then that 8 marks, you beg it. Then you have 1, right? So that is from me. Don't forget to share. Don't forget to comment. Don't forget to like. And far important thing, don't forget to subscribe. Subscribe. That is from me, Mr. Mpepu. Until we meet again tomorrow when we are dealing with our visual defects, then I'm done, guys. I'm done. Thank you for being here. Thank you for choosing me. Thank you for learning life science with Mr. Mpepu, right? So we are done. You can see today we had many questions, but it is for the best, right? Thank you for being here. Thank you for choosing us. Workers Day. If you know the best, you will do the best. My name is Mr. Mpepu, the vendor boy from Vendor. Anything else, nothing to offer you. The gold and silvers I don't have. I only have this to offer to you, guys. Thank you for being here. Thank you for choosing me. If you know the best, you will do the best. That was it from me, Mr. Mpepu. Thank you. Let's meet again tomorrow. Adios. Peace. Hasta la vista. Thank you.